So today we're going to talk about a new form of genetic encoding that we've been working on for the last few years, but now we have a lot of biological validation for. So the traditional idea of how information flows in cells is from the genotype. It gets processed into RNAs that contain codons that encode proteins that produce phenotypes. And the fun thing about multicellular organisms is there's just not one genotype. There's like thousands of genotypes potentially buried inside the genome, and it depends on which particular genotype you extract as to what the consequences are. So, it's an interesting system where when people say, are you going to get your genome sequenced? Well, that's fine, but each cell actually takes a different slice of that genome to produce outcomes that vary across tissues and across individuals. And so this is the classical view of how it works, and there's a few exceptions along the way where there's feedback from protein to how RNA is processed or how DNA is read out, or even from RNA to DNA. So, the classical paradigm has survived since the 1950s with a few kind of interesting exceptions. So what we are going to be talking about today is slightly a different way of thinking about how the genome works, and sort of in a way it's analogous to the way we divide computer into software and hardware. So, what I just talked about was all the codon that encodes proteins, and that gets translated in the ribosomes in the cell to produce machines that constitute the wetware of the cell. What we're going to be talking about is something called Fliponware, which is really a way of compiling codons to make different proteins to produce different outcomes. So we're going to be focusing on this kind of compilation that's going on at the genome level, and also occurs at RNA, but for the purposes of the discussion today, we're just going to think about how Flipons affect phenotypes and how they change outcomes, so that the same input can be processed in very different ways, and depending on the context, you get a variability in output. So we're going to start like imagining that Dimitri and Sasha, who gave a really nice presentation earlier on, applying for a job, and they applying for a job as a computer programmer. And so the interviewer says, Alex, Dimitri, what have you got to show? And they said, well, we've come up with this new programming system. It's self-replicating. That's great. It's self-repairing, so it's really resilient to damage. That's excellent. The code base is really expansive. We can add stuff in there, and we can combine them and get lots of interesting programs produced really easily. And then we can select which program that we need to do the job. And by this time, the interviewer is getting really excited. It's also, Alex and Dimitri might say, really redundant. We can be sure that we can get a particular class of responses from it, depending on how we select it. And by this time, the interview is about to give Alex and Sasha a huge check to come and join the company. But then they mentioned that this whole program they've developed is really expensive. It makes lots of errors. And actually, most of the output from that whole system is just trash. It's just junk and needs to get rid of. So there's just a whole lot of overhead in cleaning out all these mistakes. And finally, they say, well, and this is why they didn't get the job, you can run this program 1,000,000 times, but you never get the same result twice. And that's kind of the difference between the way we think about computers and the way that biology operates, that what we would do in terms of the computer code being clean and efficient and lean is exactly not what we find in biology. So we need to understand that more, because clearly the biological programs are highly successful. So let's just go into those little categories a little bit more. By expansive, what it means is that you can take any sequence of DNA, throw it into cells, and then you can change their function. You can get a gain of function, a loss of function, or actually you can make a whole new set of functions depending on what code you put in there. So imagine writing a program where you just send in a string of the alphabet there, and suddenly you can do all these new functions. Just throw it in there, and then you're able to select some combination of those letters you feed in there, and suddenly you have a whole new program. So what's more is that once you do that, and throw in those string of letters and code and all that kind of stuff, what's really interesting is that you're not limited to what you throw in. There's a whole system of combinational diversity that's generated. And this in multicellular organisms is due to the fact that the genes are encoded in pieces. So the functional part of a gene may be, in this case, in 3 separate pieces. And to actually get something that makes protein that actually works in the cell, you need to combine these pieces of the gene together and splice them. So you can combine these different functional elements that are called exons in lots of different ways and produce lots of different RNAs. And some of these are functional, some of these aren't functional, but they all lead to different protein products. So it's expensive approach because actually most of these RNAs that get spliced actually don't do anything. For example, you have to get rid of all these introns. They're not necessarily functional, so you make them, and introns can be up to 40% of the genome, and you make 40% of the genome into RNA, and then you just trash it. It's not doing anything for you, so you get rid of it. So that's expensive, and also you make lots of mistakes. You can join these exons together in a way that they don't make sense, and they're not going to do anything except introduce errors into the cell. So you got to get rid of them, and there's a whole cellular process called nonsense-mediated decay that gets rid of all this junk RNA that you make. So it's good in terms of combinatorial diversity, but it's bad in terms of the energy it takes to make sure that you don't end up introducing errors into the cell that end up destroying it. Well, it's also kind of expensive as well in the sense that you can make lots of different types of cells with all this combinatorial diversity. And here's just 4 different cells that are going, 2 are going one direction and 2 are going in another direction. They interact. And what's indicated here by these little T-shaped arrows is that each of these cells are capable of killing each other. So in fact, the way to get the response that you want involves getting rid of a lot of cells. You kind of have to kill these 2 cells here if you want to have a repressive response. And this is from the immune system. These are repressor cells. These are natural killer cells. So to get a repressive response, you need to kill off a bunch of cells. And to get a cytotoxic response, you have to do the opposite. You have to kill off the other pair of cells. And it's a kind of an intransitive logic that's involved where whichever cell predominates is determined by the context. So you can use the same cells, and by killing off some, you can change the nature of the response in a context-specific way. And again, that takes energy, it's expensive. You're making cells just to kill them because they aren't going to serve any useful function in that context. Also, it's kind of selective as well. Of all the potential RNAs you can generate and all the potential responses that you can make, you only want to select those that are relevant to the context. And one of the biggest challenges is that when in multicellular organisms that have 2 parents, you have to make sure that these genes from the different parents get along because they've never seen each other before and they don't know what each is capable of. And so you can't let one parent dominate the other parent because of the potential issues of incompatibility between the gene products. And you can just kill the organism, the embryo, at a very early stage. So you need to be able to select which of those RNAs get generated and which of those get expressed as proteins. And you can change the threshold depending on the context as well. So it's a system that makes lots of RNAs. It's combinatorial, but then the RNAs can come from anywhere. So you have to have a lot of controls and a lot of thresholds to control your responses in a context-specific way. So as I said before, when we're talking about the cells, most of the logic inside cells is intransitive. So as computer programmers, we always think of, A to B to C to E, and that if we have A, then therefore we get E. So we always think in transitive logic, and it's good for science because it means results are reproducible. It's confusing for quantum mechanics because you can have states that are incompatible superimposed with each other. But anyway, you can kind of get around that by making some additional assumptions. But actually, biology doesn't care much for transitive logic. Most of the responses are intransitive, where in this situation, E can actually dominate A. So there's a game called rock, paper, and scissors, which kind of illustrates intransitive logic, where you have two players that make a hand symbol, either for a rock, a paper, or a scissors. And so rock will beat scissors, scissors will beat paper, and paper will beat rock. So depending on what the other player plays, one response will predominate, and other responses won't. The other advantage of the intransitive system is that you can have inputs at any level that can produce outputs at any other level, where in the transitive system, you don't get such flexibility. So this transitive system is actually limits variability, which makes it programmable, and that's why we like it, because we can control the variability. But in this system here, it's built for generating diversity. So it's a different logic that's present in cells. And if you want another concrete example of it, you can think about the Krebs cycle, for example, where the carbon backbone gets passed around and around and around, and different inputs come into the Krebs cycles, and there are different outputs. And depending on the context, one input will be important, and in another context, a particular output will be important. So that's kind of captured in this slide, where not only can you input at any of these points, but you can also output a lot of these different points. And so from a biological point of view and from a computational point of view, it's really interesting. So if you actually treat these symbols here as numbers, so like in the usual sense, so from a number system like 3, 4, 5, 6, 7 in an ordered fashion, you can see that we have numbers that aren't sequential. And so from a computational point of view, that's great, because then we can make conditional logic. So we can say we will only get 5 produced if we have 2 coming in from some other source. So we can make this whole step here conditional. We can also build in redundancy into the system, where we can, if we want 4 to go to 7, we can either have 2 or 3 add together to give 9, allowing us to do that, or else we can just input a number greater than 4 into the system to get that step made. And the other thing that's fun for a biological system to do is that it can actually bypass many of these steps. So if under some condition of selection that it's optimal to go from 6 to 4 and avoid going through steps 3 to 5, you can select that process. So it's a very flexible system in terms of the logic of biological cells that we don't really capture in terms of the way that we program computationally. So a lot of this redundancy that results from this kind of cycle is actually built into biological processes. And it's, anyone who's sat through any bioinformatic large data set analysis will normally pull their hair out, because all they see are tangles of interactions and pathways, and it's really hard from a transitive point of view to try and understand what's going on. It's really difficult to kind of connect the arrows in a way that a transitive person thinks makes sense. In fact, a lot of the biological interactions are intransitive, and there are many paths from A to B, and this redundancy that's built in is based on a lot of very low affinity interactions that can arise over time and be selected, and it allows a cell to be very resilient to environmental changes, and also really resilient to errors that occur in the DNA code or the introduction of new DNA code into the cell. And I think for those who are following recent developments and thinking about how gene transcription is regulated, people are now thinking, rather than having specific transitive interactions between DNA and proteins, you actually get a lot of these condensates form, where there are a lot of low affinity interactions between cell components that contribute to A, leading to production of B. So because of all these kind of different paths that exist in the cell from getting from A to B, it tends to make the system very canalized. So it tends to, if you imagine this ball rolling down at the top of the mountain, it has a choice to go between these different valleys, and that's the only choice it really has. It can go A or it can go to B, but there's a whole other space it will never enter, because all these redundant, resilient interactions will guide it one way or the other. And this concept, was developed in the 50s by this guy called Waddington, and it's called catalyzation of phenotypes. So despite all the diversity that you can generate, one way of handling it is to build in redundancy and resiliency so that only certain outcomes will happen. And in terms of self-replicating, well, depending on which way you want to look at this, whether this guy is running away from a lion or this guy is running towards a potential reproductive partner, what happens is there's selection for variation so that survival of the species by propagating the next generation is something that is favored by natural selection. And along that process, variation accumulates from all the errors in the genome to make this process more efficient. So self-replicating computers is not something that we can make currently, but biology provides a grand example of the benefits of having a self-replicating computational system. So talking about how all this information is encoded in DNA, traditionally we talk about codons, and codons really represent the order of nucleotides in the DNA string. And so we have something that's called a DNA code based on codons, but it's actually a cipher because it just maps nucleotide triplets to amino acids. And it's just a lot of strings that are slapped together that enable you to make functional wetware. What we're going to be talking about today are Flipons, and rather than the strings of DNA, we're talking about sequences that adopt alternative conformations. And I'll give you some examples in just a moment of what an alternative DNA conformation looks like. And what the current way we're thinking about that is that this encoding by shape actually provides an instruction code for compiling transcripts from the genome. So in this combinatorial process that I mentioned before, Flipons can actually help decide which exons compile together to make a message. And so they can change the output from the genome. And because their shape is variable, depending on the context, then you can get different products from the same gene just by changing the DNA shape. So the advantages of codons, of course, is that because it's a combinatorial encoding by 4 nucleotides, you can get really high specificity in recognition of proteins and in terms of control. So you can give everything a distinct nucleotide barcode, for example, and you'll be able to specify it with high accuracy. The other consequence of using nucleotides is that you can introduce variation by mutation or some other process, and then the outcomes will change depending on what the changes that you make. The disadvantage of it, if you're going to have high specificity by having lots of variation in sequence, then getting a search efficiency going to find that particular sequence varies with genome size. And so the larger the genome, the larger the sequence block you need to be able to ensure that it's specific for a particular outcome. It's also another problem with encoding with nucleotides is that many different proteins and many different sequences are repeated within the genome, and each of these sequences or these binding specificities compete with each other. And so actually trying to find that particular sequence that you have generated with high specificity can be really challenging because you have a lot of noise from related sequences and overlapping motifs that make the search less efficient. In contrast, and all these different operations that you can do with nucleotides are described by string operations. And in biology, most of these string operations are usually one-way functions because it's actually more efficient to do that. By a one-way function, I mean that you can't go backwards. To revert back to the original sequence is kind of really challenging in a biological context. Most of the mutations that occur are imprecise. So having a mutation that changes the sequence and trying to find the exact mutation to reverse the change is pretty difficult to do. So rather than trying to go backwards, the biological solution is just to get rid of those cells where you generate errors in the DNA in the string that you can't fix. And so you basically use cell replacement to allow a system to reset. So you go back to the original genome in a new cell and start again. So most of the string operations that are done are pretty well characterized now. You know, there's clearly DNA mutation, recombination repair pathways that are error prone. There's DNA editing, much similar to the RNA editing that Sasha described. And there's also DNA transposition. So generating new strings can, within a genome, can occur by the transposition quite frequently. And that's the most common way that new strings are introduced into the genome, is having one segment of DNA transcribed into RNA and then reverse transcribed into DNA and then inserted somewhere else back into the genome. And so in humans, that accounts for about 50% of the origin of our genome, where we've had spread from one locus to another locus through this RNA intermediate and through this reverse transcription process. In other organisms, you can go directly from one locus to the other locus just by copying the DNA from locus C and inserting it in locus D. So that's when you're talking about string operations, that's an example of one type of string operation to increase the coding base for your DNA. As Sasha also introduced, there's other ways of there are other one-way string operations, and these don't act at the level of DNA, they act at RNA. So you can change the nucleotides that are present in the message by this process of RNA editing. And it can be quite extensive, like you can have addition, deletion, editing, where you get insertion or deletion of bases. And this is best well known in the trypanosomes, where their genome is completely non-functional. It's completely trash. If you read out the sequences from the DNA and try and translate them into protein, all you get is a bunch of stop codons. You can't make a protein from a trypanosome mitochondrial DNA genome. In fact, the only way you can make functional proteins is to insert or delete uridines. And so that's kind of the biggest contrast I know of, showing the difference between the way things are encoded in biology and the way you would encode them using a computer code. The other type of editing which is found in humans is the substitution editing, where you can get a change in one of the adducts on the ring, in this case from an amino group to a keto group. And so in this case, you change cytosine to uracil. And you can also do this for double-stranded RNA, which we'll talk about later, where you get the amino group removed from adenine and you end up getting inosine, which is translated as guanine if it's in a codon. So these are just examples of string operations that happen on nucleotide sequence. So in contrast with Flipons, Flipon-based logic is a lot less complicated and operations are a lot more limited, but it's a much more powerful system just because of these limitations. So there are only very few number of structures that DNA can adopt, and nature seems to take advantage of that. And so you're either in one structure or the other structure. So most of the outcomes are binary. So rather than having some kind of analog variation that you can get by varying the nucleotide sequence and varying the protein that's produced, you can just produce a 0-1 outcome. It's either bDNA or it's in some other conformation. And again, you can also have some elaboration on this scheme because some sequences can adopt multiple alternative conformations. And so you can get competition between whether bDNA flips to zDNA or flips to a G quartet or some other conformation. And we'll talk about that in just a moment. And so rather than talking about string operations, what we're talking about with Flipons and structure encoding is transition probabilities, whether basically it's a measure of how hard it is to flip from B to some other conformation. And the other difference that we have between flip-ons and codons is the whole process is easily reversible. So you can go from B to something else and back to B again. And so in that process, you can get a lot of heuristics involved where the forward and reverse rates are quite different. And you can actually vary the rates by varying the modifications that happen to the DNA. So we'll talk about that in just a moment. So here you go. This is the BDNA chromosome here, and here's some example of flip-ons where you can flip from B to Z and just by inverting the bases. And so you go from right-handed to left-handed. You can refold the DNA in a way so that's a 4-stranded structure. And this happens in G-rich sequences. And so you end up getting what's called a quadruplex. You can actually also take one DNA strand and anneal it to a BDNA helix. You can put it into one of the grooves and end up with a 3-stranded structure called a triplex. And also you can do other fancy things by varying the sequence here. You can actually introduce bends into the DNA that actually restrict its topological properties. So there's no shortage of alternative conformations that DNA can adopt. And the question always has been, what does nature do with this potential diversity and this potential variation? So here's some potential roles for these different conformations that nature can use them for. So with ZDNA, clearly it's a binary switch, B to Z to B to Z to B to Z. You can switch on and off a number of things, and I'll give you some examples later on. These G4 quartets are really stable, so they form rapidly, but it's hard to reverse the process. So potentially they're a memory element. There are other conformations that we didn't mention there that pH dependent, so it could act as the pH sensor. We have the T flip-ons or the triplexes incorporating this 3rd strand. This 3rd strand could be an RNA, so it could actually end up as a way of integrating output from a gene with the actual state of that particular gene. So if you form a triplex, maybe that's a way to indicate to the gene that it's in an active state and to maintain that active state. So these bent DNAs can act as insulators to stop topological strain from being transmitted through DNA. And then we have BDNA, which by comparison is just rich in nucleotide variation, but is not nearly as dynamic as all these other alternative structures. So we're going to actually talk mostly about ZDNA, and again, the transition is reversible and it involves just flipping the bases over, inverting the bases, and in the process, you end up changing the direction of the backbone. So instead of going from being right-handed, going from right to left, shown by the yellow strand, it's left-handed with the backbone going from left to right, as shown by the red strand. So that's the difference between B and ZDNA. It's just an inversion of base pairs. And so what we're going to talk about is the physical nature of this flip, what causes it to happen, the biological implications of being able to flip from B to ZDNA in a binary manner, and also the computational implications of having a flip-on like ZDNA. So ZDNA is, like many of these flip-ons, actually occurs mostly in simple repeat sequences where you have a preferred sequence that will go from B to Z. And for example, in this sequence where you have alternating C and G, why that is preferred is because the ZDNA actually has a dinucleotide repeat in the backbone. The dinucleotide repeat is associated with the structure of the backbone, and we'll talk about that in just a moment. There's also other sequences that are repetitive, such as the ALU sequences that were copied through RNA from one position in the genome to another position in the genome, and they're about 12% of our genome. So they also have Z forming elements. And also endogenous viruses end up forming Z DNA or zRNA just because of all the topological problems they have in packaging themselves into protein capsules. So interesting thing about all these repeat sequences is that they can be spread through the genome relatively easily, either through recombination or through reverse transcription. And the effects on the readout information at the new location is completely is locality specific. So they change the readout at that particular location. So you can spread these flip-on sequences throughout the genome, and then you can select the particular flip-ons which changes the readout from that locality to give you a beneficial phenotype that can be selected by natural selection. So they're very active elements of the genome, changing shape. They spread around the genome, and they're also genetic elements because they are subject to selection because they affect phenotype. So part of the physics of, or physical chemistry of B to Z transition. So zDNA is a higher energy state. It takes energy to flip the bases over and to form zDNA. And as I said before, it's a transition that doesn't normally follow the same forward and backwards pathway. So it can takes the time to form zDNA versus the time to relax zDNA is different because of this ketosis that occurs here. So there are different things that can affect the energetics of this transition. So you can put base adducts there that change the propensity for bases to want to flip over. And you can also put energy in there in a number of ways. And one way is supercoiling, and we'll describe that in the next couple of slides. So here are potential base modifications that can occur on the rings. So when we talk about adducts, we're talking about modifying like cytosine or guanine, for example. And there's a whole variety of different modifications that occur which can increase the propensity to form zDNA by lowering the transition energy. And amongst these well-known 5-methylcytosine and all the oxidative derivatives of 5-methylcytosine that also affect the potential of bDNA to flip to zDNA. In terms of guanine, we can get oxidative modifications such as that affect the ring position, and they also increase the propensity to form zDNA and also G4 DNA. So in some ways, in those particular contexts, these base modifications allow a cell to sense that there's oxidative damage occurring and form an alternative conformation that can activate particular biological repair processes. So in terms of supercoiling, this is something that happens in covalently closed circles of DNA. So if you imagine this is a double-stranded RNA like a plasmid, and it's covalently closed, there are no gaps or anything like this. And so it's twisted around itself a little bit. And so one way to relieve this twist is actually by taking the segment of bDNA and flipping it to zDNA. So you go from right-handed bDNA to left-handed DNA, and you absorb these writhes and relax the plasmid. So that provides energy. The energy of the supercoiling provides energy to stabilize the zDNA conformation. And what's kind of interesting, and you'll see why in a short time, is it takes about 1.65 negative supercoils to flip this bDNA into zDNA. And about 1.6 of that is the actual difference in the helical parameters of bDNA and zDNA. But also you have to allow for the fact that there has to be BZ junctions, the junction between left-handed and right-handed DNA, that causes some unwinding as well. So as I mentioned, switching the shape from bDNA to zDNA requires power. One source of that power is RNA polymerase. As it goes down the double-stranded DNA, it unwinds the double-stranded DNA so it can make RNA, and that unwinding causes negative supercoiling, which can power the flip from bDNA to zDNA. Another way of doing it is actually by stretching DNA. So if you have a helicase that stretches the DNA, so it pulls on the end, that also introduces tension in the DNA, and you can relax that tension by flipping from B to zDNA. And the reason the tension is relaxed is because BDNA is 34 angstroms long and ZDNA is 45.6 angstroms long. And so by doing the flip, you can make the DNA longer. And so when you try to stretch it, flipping it to ZDNA relaxes the tension that you generate. And this can be shown with single molecule measurements with high degree of accuracy, and all that energetics is well quantitated. The other interesting way of doing it is normally our DNA is wrapped up around nucleosomes, and that allows it to be really compact. But if you flip out the nucleosome and just lift with DNA, you can then generate these negative supercoiling, because the negative supercoiling is constrained by this solenoidal wrapping. But when you flip out the nucleosome, suddenly you have all this negative supercoiling generated, and then that can provide the energy to stabilize ZDNA. So there's a variety of different ways that you can flip a sequence from B to Z. So that's all fine in theory, but what about the biology of ZDNA? We have all these nice physical chemical measurements, and we understand the energetics with great precision, and we understand all these different aspects of flipping from B to Z. What about the biology? And so the biology of the ZDNA actually has been unlocked by the identification of the ZDNA specific domain, the Z alpha domain. And this Z alpha domain is structure specific. It doesn't recognize any sequence specific features of ZDNA. It just binds to the backbone. And you can see the zigzag nature of the backbone that relates to this dinucleotide repeat that I talked about before, because these sequential bases have a slightly different conformation around their sugar, and that different conformation causes the backbone to be zigzag, and that's why it's called ZDNA. And so Z alpha binds to ZDNA in a very highly specific manner, and it was possible to unlock the biology of ZDNA by using the Z alpha variants. And so there are a couple of residues that make very specific contacts with ZDNA, and by binding variants in these two bases here that exist naturally in humans, and looking at what kind of phenotypes they produced when people had those two variants in Z alpha domain, it was possible to understand what Z alpha is actually doing in the cell. And from that, then it's possible to deduce that ZDNA is actually the functional element that Z alpha responding to, and is actually controlling the phenotypes that were observed. And these phenotypes were to do with regulation of interferon production. And so if you have the variants, you tend to make a higher level of interferon than if you don't have the variants. So in that sense, the Z alpha domain was turning off the interferon responses, a negative regulator of interferon responses in normal people, but not in people with the variants. So the Z alpha domain was isolated in this enzyme that Sasha introduced, this enzyme called ADA, which is a double-stranded RNA deaminase, and it has a Z alpha domain which binds ZDNA and zRNA, and it also has double-stranded RNA binding domains which are structure-specific and not sequence-specific, and it has this attached to a catalytic domain that performs the editing function. So it's an example of where you're mapping shape functions to string functions. So you have the Z alpha that binds to ZDNA, which localizes the enzyme. You have the double-stranded RNA binding domains that bind to fallback structures and RNA that form the double-stranded RNA substrate. And then you have the string operation where you have the editing of adenosine to inosine to produce change in the base at that location. So it's a nice example of where you can use shape and sequence, and you can use shape functions and string functions to produce an outcome. But how does ADA prevent the immune response from happening? So it turns out that there are host sequences in double-stranded RNA that have a propensity to form zRNA, and these sequences are present in host RNAs, but not in viral RNAs, for example. And so when you initiate the response to double-stranded RNAs, which is a natural response because most viruses make double-stranded RNAs, and so that's a warning sign that something's wrong in the cell. So if you get double-stranded RNA being produced, you want to know whether it's a host double-stranded RNA or a viral double-stranded RNA. And so as the response is activated, you have these helicases which then combine the double-stranded RNA, and they actually stretch the double-stranded RNA. And if the host sequence is there, that forms the zRNA. Some sequences will flip to zRNA, identifying it as a host sequence. Then ADA will bind through the alpha domain to those host sequences, and then we'll perform the string operation to make edited RNA and get rid of this double-stranded RNA that's causing the problems. Of course, because viruses don't have these Z-forming sequences, then they remain as double-stranded RNA and can invoke interferon response. So it's just a nice example of where you can combine shape and string functions together to produce a biological outcome. So let's talk about the Flipon computational gates. What can you do with Flipons with this binary 0 or 1 state? And so you can use Flipons for logical operations, and I'll give you some examples of AND or NOT gates that we can make with Flipons. You can use them as topological switches that allow you to have action at a distance, and you can use them as state switches where you can switch something on or off, or you can toggle it between states. So here's a logic implemented with Flipons using RNA splicing. So as I mentioned, you can have alternative splicing that happens. So you can have either inclusion of exon 2 or exclusion of exon 2. So you get alternative products produced. So, and you can have double-stranded RNA that brings the different junctions together that enable splicing. And then you can have double-stranded RNA editing happening, which changes the probability of exon 1 joining to exon 2 versus exon 1 joining to exon 3. So here you have the splice is conditional on Z formation and on binding of the P50 Z alpha containing 801. So if you have both these Z formation and you have the presence of this editing enzyme in the cell, then you can edit the double-stranded splicing substrate and change whether exon 1 goes to exon 2 or exon 1 goes to exon 3. So how can that be turned into a NOT and an AND gate? Well, imagine that exon 2 is actually a kill tag. So if this is present in the RNA that is produced, then that RNA gets destroyed. So that automatically gives you a NOT gate. So if you join exon 1 to the kill tag and then to exon 3, that RNA will automatically be trashed. So you have a NOT gate. So any RNA that has this kill tag will get destroyed. On the other hand, if you have exon 1 connected to exon 3 directly, then that acts as an AND gate. So being able to use Flipons to change the outcome of an RNA splicing process is quite easy to do, and it just illustrates how Flipons can control this particular inclusion of this tag. So the Flipon, by excluding this kill tag, makes an AND gate. By including this kill tag, makes a NOT gate. So considering that you have potentially millions of these Flipons throughout the genome, then suddenly you can start thinking of the genome as a binary genome, and you can start thinking of all these potential AND and NOT gates that are through the genome, and you can start thinking of all the combinational diversity you can generate, and all that diversity changes phenotype, so it can be selected. So it's, rather than thinking of an analog sequence-based genome, you can think of a genome full of Flipons and binary switches that can change outcomes. So another example is the topological switch, and this alludes to the fact that you can have a sequence of Z forming that when it gets flipped from B to Z, it changes the topology of the whole domain, and that change in topology gets propagated along the DNA and allows action at a distance. So flipping this particular sequence here from B to Z may change the conformation of a sequence far, far away and flip that back from Z to B. So this allows you to have a topological switch where you have input at this particular sequence, changing the output of this particular sequence. And this is occurring over large distances of DNA. So it's an interesting feature of chromosomal organization. And so depending on what sequence is Z forming at each of these two sites. In this case, we have a very good Z former. By that we mean it's easy to initiate the flip because of its sequence composition. And we have another Z former here, which doesn't actually flip very well, but once it starts flipping, it's long. And so it actually can capture all the topological energy. And the consequence is that when you start talking up this particular closed circle double-stranded DNA, you initially induce the flip in the Z1, C8 sequence, but as you keep cracking it up, suddenly you flip the CA sequence into Z, and this absorbs all the topological energy and flips the Z1 sequence back to B while capturing Z formation in the Z2 sequence. So it's not only a theoretical possibility, but you can actually look at this in gels and actually demonstrate that these two different conformations, these two different Z forming sequences compete with each other to form zDNA, and that you can switch zDNA from one to the other just by changing the amount of super helical density in this particular closed circular plasmid. So that's an example of a topological switch. You can also use wrapping of DNA into these nucleosomes that were mentioned before. So normally, because the genome is so large and because a lot of it is non-coding and of dubious value, most of the DNA is actually packed into these nucleosomes, and so this represents an off state. So they're tightly packed into a dense bundle, and this is called heterochromatin. So this heterochromatin exists, but it's not transcriptionally active. It's not contributing to the codon wear in the cell. So it's definitely in an off state. So the wrapping in histones progresses any transcription. So you can use this actually as another source of Z formation, as we mentioned. So if you eject this particular nucleosome, then you flip potentially that sequence into zDNA. And it just so happens that, again, we come back with this magical number of 1.6 turns around the nucleosome is actually sufficient to flip one vDNA helix to a zDNA helix. So this is another way of using histones to regulate gene expression and also to regulate flip-on conformation. So if you start thinking of nucleosomes like in this context, you think that the nucleus has a whole bunch of energy stored in these conformations and these structures that is waiting to be unlocked. And if you can unlock that energy, then you can stabilize these alternative DNA conformations and you can use it to do interesting biology. So this can be used, for example, to eject the nucleosome, and this generates energy to generate open chromatin, and then this can be used to actually then this energy that's stored here then can be used to assemble the different transcriptional factors that are necessary to guide readout of that particular chromatin region. And so here zDNA is just acting as an energy store, so it captures the energy from unwrapping the nucleosome, and then it uses that energy to help power the assembly of these enhanceosomes. And even though we're just talking about one factor here, these enhanceosomes themselves have structure and they involve a lot of negative supercoiling of the DNA and wrapping it around different components. So this, in this case, the flip-on acts as a store of energy to enable this transition from one state to another state. And also you can change out one particular enhanceosome binding factor to another just by using the same mechanism. So you can kick this off, capture the energy as zDNA, and then use that energy to stabilize a new conformation there. So just to compare the different types of logical operations, state versus sequence, you can see that sequence at high specificity, finding it is search dependent. The outcomes change with sequence variation, and there's competition between the different target sequences that you're searching out. In terms of structure, it's much less complex. You get binary outcomes, and you get confirmation between and competition between conformations. So there's lots of variations on the themes there. You have other Flipon types, you have the string to structure functions that I talked about, and then the search algorithms that are involved that underlie finding information in the genome. So the G quartets are these four-shaded structures. They form the site suburban chromatin, and they're associated with gene regulation and DNA repair, and they're very stable once folded. Triplexes allow you to put a third strand into bDNA, and that helps localize whatever's attached to that third strand. And so that third strand can have lots of different potential motifs that can be recognized by proteins, and so that you can get a lot of combinatorial diversity in the proteins that you actually attract to that site of triplex formation. So that's another example of how you can use an alternative conformation to localize cellular machinery to a particular genomic site. You can use strings to create structures, and so you can have complementary strands that base pair with each other, and they combine to form a structure. And why is that useful? Because it allows you to use structure-specific proteins to localize that have particular patches on them that allow you to localize transcriptional machinery to that particular structure. So the genetic code is a fine example of this, where you have core codon machinery that reads out the RNA from the genome, and you have core translational machinery that takes the RNA and converts it to protein. But it all depends on actually making a shape between the nucleotide triplet and a tRNA for this particular core translational machinery to engage. So using generic machinery is possible just because you actually convert the sequence into a shape that's recognized by the generic machinery. And this is not only true of the genetic code, but it's true of other regulatory RNAs, where the adapters can be microRNAs that control RNA stability, or they can be guide RNAs that control CRISPR-based immunity. So here's an example of just going from string to structure to function. So the last thing I just want to cover is the type of strings that are called repeats. They're very frequent in the genome. They are low complexity. They repeat the same sequence, like poly A, poly G, alternating CG, etc. They don't have much information in them because they have low complexity, but they're actually very useful for facilitating search of information in the genome. And so these low complexity sequence repeats can make structures, as we've been talking about, that make Flipons. They can actually tag RNAs, so that you can tag a bunch of unrelated RNAs with the same particular low complexity sequence, so that they can be co-regulated. And they also encode peptide patches on proteins, and that can also help these proteins find other proteins that may be necessary to help assemble a biological machine. So each of these different types of outcomes from simple sequence repeats allow search. So you can search by structure, you can search by target, and you can search using these patches, these peptide patches on proteins. And so the indexing varies by where these sequence repeats act, whether they act on DNA, RNA, or protein, and the location within DNA, RNA, or protein actually determines whether they are active or not. And you then have natural selection to maximize the efficiency of the search function in terms of trying to find a particular location or particular target, or to build a particular biological machine. So we haven't mentioned peptide patches, but they're kind of really interesting. Peptide patches made by low complexity sequences enable the assembly of lots of different biological machines, and this is the way they work. These low complexity structures actually don't actually fold into anything. They can have lots of variability in their conformation, and they can wave around searching for things in their immediate environment. It's just like putting a huge set of tentacles out to find something that's of interest. And when they find something of interest that's complementary, they can fold, as seen here for this protein, and into a biological machine that performs a function. So these peptide patches, these low complexity sequences that don't fold, actually enable search between different proteins to enable whatever function in this protein has to be brought to in close proximity to whatever function this protein has without actually affecting the protein function at all. So these are just tagged on to the core functional domain, and they're just low complexity regions that help these different functional domains find each other to make a machine. So that's it. So I convinced you that Flipons are a great way of encoding genetic information. They change how codons are used in the cell. They change how the wetware is assembled in a cell, and because they change phenotype and change biological function, they're subject to natural selection. So then actually elements of genetic elements of a multicellular genome. So thank you very much for your attention. I'm